Hi everyone, welcome to our new lecture. So today we're going to be talking about soil microbiology and biogeochemistry. Let's jump right in. So firstly, let's start off with what biogeochemistry is. So biogeochemistry is the scientific discipline that involves the study of chemical, physical, biological, and geological processes and reactions that govern the composition of the natural environment. So you can think of it almost as the cycling of different compounds, or the cycles that how different compounds move throughout the environment. So the study will then include the biosphere, the cryosphere, the hydrosphere, the pedosphere, the atmosphere, and the lithosphere. And so just in summation, essentially, biogeochemistry is the study of the cycles of chemical elements such as carbon and nitrogen and their interactions with and the incorporation into living things transported through the Earth scale biological systems in space and time. So it's a very, it's a mouthful, but to sort of simplify it for you guys, essentially, it's just how different chemical elements move through the environment into and out of living organisms, and how these organisms will alter these these chemical elements as they move through the environment, as they utilize them, or as they become waste products. So thus, you can also think of it that this field will focus on chemical cycles that are driven or influenced by biological activity. Of course, if the microbes that require or that cycle these nutrients are more abundant or less abundant, that would impact these chemical cycles. Particular emphasis is placed on the study of carbon, nitrogen, sulfur, iron, and the phosphorus cycles. We won't be discussing all of those, but we will be discussing quite a few of them. So let's first look at soil and the role it plays in the global carbon cycle. So soil is actually the largest terrestrial reservoir of carbon. The amount of carbon stored in soil depends on two factors. Firstly, it's the amount of carbon entering the soil. So this is typically from plants, or the amount of carbon leaving the soil following decomposition reactions. And this is often driven by microbes. And so the rate at which the carbon will either enter or leave the soil is dependent on quite a few factors. First would be temperature. As we know, and we've discussed many times in this course so far, temperature will impact the rate at which chemical reactions can take place. So colder temperatures, you will find that chemical reactions typically slow down, and in warmer conditions, the chemical reactions will typically speed up because the molecules are just moving more and more collisions, more chance for reactions to take place. Soil moisture, similarly, we know how important water is in metabolic processes. So soil moisture will also impact the rate of carbon leaving the soil when microbes are decomposing, dead organic matter or inorganic matter. But obviously, if you're talking about carbon, it has to be organic matter for the most part. And then plants, the metabolism of plants will also be subject to soil moisture. So moisture will impact the amount of carbon entering and leaving the soil. Soil mineralogy, this will impact the types of microbes and the type of plants that can grow in your area, for example. So that could definitely impact how the rate of CO2 flux. And then the microbiology of the soil, of course, because microbes are what drive the decomposition process for the most part in soil, or at least play a vital role in that process. You definitely need to account for that when looking at the flux of CO2. So this is actually a diagram. I see now that it's not really playing, but this is a website that you guys can visit. It's a model produced by a NASA supercomputer that shows greenhouse gases like CO2 and how they fluctuate throughout the year. And you can see the red representing, or the more brighter colors, reds, yellows, and oranges representing higher carbon concentrations, and how those are sort of centered around North America, Europe, and then of course also East Asia. You can specifically see when you play this, because this is actually a moving model, so you can actually see how the flux intensifies and how this top section essentially becomes bright red. Sorry. Becomes a bright red color just illustrating how much CO2 is actually produced in the northern hemisphere. But something that we also have to keep in mind, CO2, there's a lot of focus on that because it's a greenhouse gas that humans are definitely contributing to a large portion. But if you look at something like saprotrophic microbes, so the microbes that decompose organic matter, they release 55 billion tons of CO2 a year, which is 8 times the amount that humans are emitting to the atmosphere. So it's not solely humans that are responsible for releasing carbon dioxide into the atmosphere. It's just that we are increasing it or releasing it at a very unnatural rate, and the amount that we're releasing is increasing over time. So it's definitely a concern. But historically, scientists have actually treated environmental microbial communities sort of as a black box. So what does that mean? So first, let's define what a black box is. So in science, computing, and engineering, a black box is a system which can be viewed in terms of its inputs and outputs. So here you have environmental conditions and your biogeochemical cycling. So there are your inputs, you feed in your environmental conditions, and then your result or your output will be your biogeochemical cycling. One of the key characteristics about a black box is that there is no knowledge of its internal working. So you only have the group of inputs and outputs. You have no idea what's going on over here. The reason why we, or the scientific community typically treats this as a black box is because microbial communities have been very, very difficult to study, historically at least. But just because they are very hard to study doesn't mean we shouldn't at least try. Because, I mean, at this point, we're leaving out the attempts to connect environmental conditions with ecosystem processes such as, rates such as biogeochemical cycling. So we don't have a very in-depth understanding of this middle black box portion, or at least historically, that has been the case. But anyway, so in our attempt to understand this, we have to try, because inside of the black box are some of the Earth's most diverse and complex communities, these microbial communities that we've been discussing up till now. So for instance, a gram of soil typically contains thousands of taxa from all 3 domains. So that includes bacteria, archaea, and eukarya. And the reason behind this diversity is the fact that soil creates multiple microhabitats for these microbes. So consequently, different microbes will outcompete each other in different areas of the soil, leading to diversity. So you might have a portion of let's say your handful of soil that has a bit more moisture. So microbes that are more well adapted to exploiting this moisture will outcompete the others. But in a different section of the same little handful of soil that you have, you might have a drier area. Thus, those wet-dependent microbes, if you will struggle to compete with other microbes there, so they will be less abundant there. So there will be this diversity, because, I mean, you can look at moisture, you can look at nutrients, you can look at organic matter concentration. There are quite a group or a bunch of variables that create different niches or different microhabitats for these organisms to inhabit. So if you look at another example, a good example is if you look at soil organic carbon, or SOC. So if you look at studies have found that if you look at the soil organic carbon quality and soil aggregation, they create distinct niches for different microbial communities, much the same as the moisture example I just discussed. So for example, soil organic carbon associated with macro aggregates promotes microbial diversity and community structure, which are distinct to microbial communities associated with soil organic carbon of micro aggregates. So it's, again, we've discussed how quickly these organisms grow and how well they're adapted to exploiting different conditions and adapted to live in different conditions. Thus, you can imagine in this handful of soil where there are so many different factors on that micro or on that small scale. Micro is not the right word. On that small scale, you can just imagine how many different little habitats there are. If you wanna look at it as just keep in mind, this is just an example to give you guys an idea. So you can almost view it as in this little amount of soil, you will have savanna habitats, you will have forest habitats, you will have desert habitats on that microbe scales. Thus, you will have just an immense amount of microbial diversity in that little bit of soil. Okay, anyway, moving on. So to just hammer that point home, there are millions of bacteria per gram of soil. And that would obviously include thousands of species then per the gram of soil. But here's the kicker. Up to 99% of them cannot be cultured. And I want you guys to reflect on that and think why that is. Just take a few seconds. I'll give you some hints. Okay, so the primary reason why these organisms can't be cultured is we cannot oftentimes recreate the conditions that they need to survive in the lab. They are dependent on nutrient conditions or climatic conditions that we simply cannot emulate, or we don't know how to emulate yet. So we can't culture them. But something that I do want to tell you guys here is that previous studies have demonstrated that there are remarkable changes in microbial community composition with soil depth across different environments. So typically, there's a consistent decrease in microbial abundance and diversity with increasing depth. So why do you think that is? So essentially what I'm saying here is that the most microbes that you find, or the most diversity and highest microbial numbers you'll find, will typically be in the topsoil. So why would that be? And the answer is simply because that area is so rich in organic material. So there's just an abundance of energy sources that these microbes can exploit. And here's a good example of what we just discussed earlier about the so-called little savanna areas, little forest areas, little desert areas, or the different niches or the different areas that form. So in this little clump of soil that we have here, and this is very, very small because that's a little quartz crystal over there. There's an air pocket, there's some water over there. But you can just see how different it is. Like the microbes growing, the microbial colonies growing on this quartz crystal here have completely different conditions that they're exposed to than the microbes growing on this clay particle next to the water, or the ones that are on the quartz that are next to the organic matter, or the quartz that has this little housed air pocket, if you will. So there are so many different little niche areas, and the microbes that are dominant here will not necessarily be dominant there, and they will not necessarily be dominant there. So different species will predominate different areas, thus you have diversity. So how do we measure this diversity? So, or at least how do we measure the microbial composition and the diversity? So I just decided to include this slide. This figure should hopefully ring some bells for you guys at this point. Just as a little sort of a callback here, remember back to the usefulness of our 16S rRNA. Remember that this molecule have both conserved and variable regions, so they're perfectly suited to measuring the similarity among different microbes. So first, what you would have your soil sample from which you would extract DNA, and you would then specifically amplify this 16S rRNA gene if you are looking at microbial community composition and diversity. Then you would move on to sequencing. Then you would, from the sequencing, you would obtain cluster OTUs, and OTUs are operational taxonomic units. So these are essentially the closest thing to bacterial species. Essentially, the term OTU is used in a different context and refers to clusters of organisms grouped by DNA sequence similarity of a specific taxonomic marker gene. Okay, that's a mouthful. Essentially, what it says there is the different organisms, the different microbes, are essentially just grouped together by how closely related or how similar their genetic material is to one another. So, in other words, these OTUs are pragmatic proxies for species at different taxonomic levels in the absence of this traditional system of biological classification that we have for macroscopic organisms. So, for several years, these OTUs have been used most commonly with units of diversity, especially when analyzing this 16S rRNA and the small subunit of the 16S rRNA in the data sets. And then sequences can be clustered according to the similarity to one another. Again, this is how related they are to one another. In the LAC, we don't have species for these organisms, so we're just assigning how close they are to one another based on their genetic similarity, just to reiterate that. So then these sequences are then clustered according to their similarity to one another, and that's what that looks like. We're clustering them together here. And the operational taxic units are defined based on the similarity threshold. So this is typically a threshold that the scientist who is heading the investigation or writing the article, whatever, decides upon what level of similarity would constitute closely related organisms. Usually that is set at 97% similarity. However, it could also be higher, or in rare cases lower than that too. Typically in sciences, we like to refer to previous peer-reviewed article. So if there is an accepted threshold of 97%, that is what most people will end up using. I do want to note here, however, that although this makes a pretty picture and it looks perfect, there is still some debate among scientists about how well this commonly used method actually recapitulates true microbial species phylogeny and ecology. So there is still some doubt in the system. Doubt is a strong word. There is still some uncertainty is a better word. Where some scientists still feel that we need more information, or we could potentially have more information. Okay, so following the clustering of these operational taxonomic units, we would then go to analyzing the community. So here we would look at diversity questions, including questions like which organisms are present, how many different organisms are there in our sample, and how are they related. So this, you could then arrive at something that looks like this, where you have an indicator of which organisms are present, you have a indicator of how many different organisms there are, and then how related they are to one another based on the similarity of their genetic sequences, of course. And then you would look at also community composition. So what are the proportions of the different groups of organisms? Here you can have a key, and then you have different, this is a freshwater sample, this is a saltwater sample, and you can now see how the microbial community will differ based on it gives us some insight into the reason behind the differences. Are these organisms then more well adapted? If you look at this dark green group over here, why is there so much more here than there is there? Might they be more well adapted to the salt environment than to the freshwater environment? And then if you look at nitrospirae, it's the inverse, where you have this big percentage of them occurring in this community, yet when you move to the salt community, they're much less abundant. So this gives you some insight into the reason behind the differences, especially if you then measure other climatic differences. You can also do, this is just an example of freshwater versus saltwater, but you could also do anything here, different temperatures, different nutrient compositions, different soil types. There's a lot that you can do here to look at the differences and then see which microbes dominate which habitats, and then perhaps even get some insight into why that might be. Okay, so why do the communities matter? And that essentially just boils down to because the different operational taxonomic units, or OTUs, have different activities. They play different roles in the environment. They do different things. So this is an example of looking at something like decomposition. Here we have different communities across, affecting ecosystem processes such as decomposition, where you have a control, which is typically an unaltered or a natural condition when you're looking at these types of studies, and then you have a drought, an area that would lack moisture. I'm not sure if this was, I'm assuming this was some soil sample, and but I'm not sure, so don't quote me on that. And then this here, you would measure the mass loss. This would typically be a percentage of your organic matter that is lost over time. And here you give your time on your x-axis. So here you can see the control. You have a greater amount of mass lost over time than you have in your control group. And this is because your microbial community that is responsible for decomposition is being affected by the lack of moisture. Okay, so that gives you an idea. Okay, guys, you're going to have to bear with me here. This is a pretty long slide. I have quite a few things, quite a few notes that I have to go through. But just try and keep up. It's not really that intense, but okay. So the primary biogeochemical cycle that we will be focusing on is the carbon cycle. And here you can see a representation of this carbon cycle. And essentially in this cycle, all organisms, including plants, microbes, animals, anything, contains a large amount of carbon in the form of organic compounds, such as cellulose, starches, fats, proteins. All of them require carbon. And I want you to recall here that autotrophs perform an essential role for all life on Earth. Keep in mind that this is not only plants, some microbes do, which you are well aware of at this point. They reduce carbon dioxide to form organic matter. So when you look at a tree, if you look at this little tree over here, you might think that its mass is from the soil where it grows. But in fact, its great mass of cellulose is derived from the 0.03% of carbon dioxide in the atmosphere. So that is where it gets its mass from. It's because it is essentially turning carbon dioxide into organic compounds, and that is how it puts on mass. That's why, that's how it makes organic material. So this occurs as a result of photosynthesis, the first step of the carbon cycle, in which photoautotrophs such as cyanobacteria, green algae, plants, and even green and purple sulfur bacteria fix. So fix, you can also think of it as meaning incorporate. So where these organisms fix carbon dioxide into organic matter using energy from the sunlight. So fixing can also be incorporation. In the next step of the cycle, you will then have chemoheterotrophs, such as animals or protozoa that eat these autotrophs and may in turn be eaten by other animals. You know, just think of a normal food chain and with the different trophic levels. Thus, as the organic compounds of the autotrophs are digested and resynthesized, the carbon atoms of the carbon dioxide are transferred from organism to organism up the food chain. Again, just think of those trophic levels, your little food pyramids. The chemoheterotrophs, including animals, use some form of organic molecules to satisfy their energy requirements, hence they are heterotrophs. When this energy is released through respiration, carbon dioxide immediately becomes available to start the cycle again. So you can see how that turns a little loop. The carbon dioxide is used by the plants to make organic matter, which is then eaten all the way up the food chain. And when respiration happens, you have carbon dioxide being put back into the atmosphere, which the plants can then use again, and that is how your cycle will work. So much of the carbon remains within the organisms until they excrete it as waste or they die. When the plants and animals die, these organic compounds are decomposed by bacteria and fungi. And during decomposition, the organic compounds are oxidized and CO2 is also returned back to the cycle. So there you have even more CO2 coming back into the atmosphere to be used by the photoautotrophs to start the entire process again. So as a note here, remember, decomposition and respiration releases carbon dioxide, and specifically decomposition releases soil carbon, very, very notably. And then photosynthesis fixes carbon, so it removes it from the atmosphere, it incorporates it. And I want you there to think of these autotrophic organisms that we have discussed. Okay, so moving on to the methane cycle. So the methane cycle actually forms part of the carbon cycle. And it's actually a pretty interesting aspect of the carbon cycle because it deals with CH4 or methane gas. And methane is a very, very potent greenhouse gas. It's 25%, it has 25 times, not percent, 25 times the warming potential of CO2. So there's a lot, it can really heat up the air a lot, which we'll get to. So methagenic archaea, methanogens, by their name, you can think what they do, are microorganisms that produce methane. Methanogens, genic, meaning produce. And they produce methane as a byproduct. They're strictly anaerobic, and they can be either autotrophic or chemoheterotrophic. And there you can see what hydrogenotrophic methanogenesis will look like. And this is, you can see the acetoclastic methanogenesis will look like this. But here's the kicker. Sediments on the ocean floor contain about 10 trillion tons of methane, which is essentially twice as much as the Earth's deposits of fossil fuels such as coal and petroleum. So it's a lot. It really is a lot. Furthermore, methanogenic bacteria in the ocean's depth are constantly producing more. They just like keep pumping out even more and more and more methane. And because of how potent methane is as a greenhouse gas, the Earth's environment would be ridiculous. It would be dangerously altered if all this gas escaped to the atmosphere. So that's why the methane cycle is so important to look at. So this is a breakdown of the methane cycle. You can see there's our CH4, and then it will split in all directions, and you'll have this little cycle continuing, or happening at least. So first, methanogenesis or biomethanation is the formation of methane by microbes known as methanogens. So here you have methanogens that are in the soil, and they actually produce CH4. Organisms capable of producing methane have only been identified from the domain Archaea, specifically a group of phylogenetically distinct from both eukaryotes and bacteria, which you know. And although many live in close association with anaerobic bacteria, pretty notably. And the production of methane is an important and widespread form of microbial metabolism in anoxic environments. It is the final step in the decomposition of biomass. We'll be looking at that in the next slide, so just keep that in mind. And methanogenesis is responsible for significant amounts of natural gas accumulations, the remainder being thermogenic. Okay, now, so we've discussed methanogens now. So what in the world would be a methanotroph? Well, as the name suggests, methanotrophs, or they're also called methanophiles, are prokaryotes that can metabolize methane as their source of carbon and energy. They can be either bacteria or archaea and can grow aerobically or anaerobically, and require the single carbon compounds to survive. So here you can see the methanotrophs and how they take in CH4. So methanotrophs are especially common in or near environments where methane is produced, although some methanotrophs can oxidize atmospheric methane. So here I want to point out two things. On this diagram, note how this methanotrophs, yes, they can take in, some of them can take in atmospheric CH4, but others also wait for this plant decay in natural wetlands and rice paddies. Because remember, as I just said, that some of these organisms, their metabolization of methane forms the final portion of decomposition. Again, we'll look at that in the next slide, so don't freak out about too much. So essentially, their habitats will include wetlands, soils, marshes, rice paddies, landfills, aquatic systems, and more. Anything that almost anywhere you can think of, actually, where you will have access to methane. There are special interest to researchers who are studying global warming, as they play a significant role in the global methane budget. Because again, they remove this methane from the atmosphere, which is good for us because that would slow down the rate of the greenhouse effect. It would slow down climate change. And it would also then, yeah. Okay, so here's our methane cycle that we can look at. I want you to just quickly walk through the diagram. I'm not going to go in any great depth here, but so first you will find complex organic matter, and hydrolysis will take place. Now you have simple organic compounds. So now you have oxygen or NO3 reducing bacteria, or iron and Mn reducing bacteria, which release carbon dioxide when they decompose or utilize these compounds. The simple organic compounds can also be broken down by fermented bacteria. There's that process, not going to focus too much on that. Now you have these alcohols, the fatty acids, the acetate and the H2, and then you have acetogenic bacteria, which can utilize those, forming acetate. Here's the kicker. Here's the important portion where I wanted to get to. The acetate can then be broken down by methanogenic bacteria, and that will yield the methane. Here's the thing, though. This is the final portion of the decomposition. You have decreasing energy yield, as you might imagine, if you're moving from these complex organic materials to bonds containing very limited amounts of carbon. or molecules containing limited amounts of carbon, I should say, you have a decrease in your energy. So this is way more energy efficient and energy rich that happens here. And yeah, so where you find the methanogenic bacteria here in the bottom right, I want you to note its location, because methanogenesis is the final step in the decay of organic matter. So during this decay process, electron acceptors such as oxygen and ferric iron sulfate, so here you have examples of that. There's your oxygen, you have ferric iron, there's your sulfate, and here's your nitrate. They become depleted while hydrogen and carbon dioxide accumulate. So these release carbon dioxide, but yet the final one will release methane. Okay, and just a reminder here, with this fermentative or fermentation, just remember that fermentation is the chemical process by which molecules such as glucose are broken down anaerobically. Thus, when we arrive at fermented bacteria, we're typically no longer at the soil or water surface. Just so that should give you an idea of where you might find the bacteria in this section here. These will typically be subsurface bacteria living deeper in the soil profile. Not very deep, but deeper, as opposed to the ones that you would find on the surface. Okay. So just again, this is just a simplified breakdown of what that process looks like. I'm not going to spend too much time here. Just again, a reminder that methanotrophs or methane oxidizing bacteria are prokaryotes that are able to metabolize methane as their only source of carbon and energy. This is what that process looks like. You would, of course, start with CH4, and then you will keep going through this process until you assimilate the carbon as you require. So there's a carbon, there's a carbon, there's a carbon that would all become assimilated into cellular material or metabolites. So yeah, just keep that in mind as you study for the exams. So now I think it's worth talking a bit about, you know, what would be some consider the first American experiment in microbiology. So in 1783 in Rockingham, New Jersey, residents alerted George Washington and Thomas Paine of swamp flares. So essentially these swamp flares are, you can think of them as these little glowing lights that would sometimes form over bogs or swamps. And people like it, what typically would happen is as people would approach these lights, they would seem to fade away or move away. And that is, and the question was, what is the source of these swamp flares? And they were also known as will-o'-the-wisps, or foolish fire. And so it almost had this appearance of this atmospheric ghost light that was seen by travelers at night, especially over bogs, swamps, and marshes. And it resembled a flickering lamp, and then is said to recede if approached, drawing travelers from the safe paths, because they would then follow these little lights. As we've learned from so many movies over the year, never go into the light, guys. So the phenomenon is known by a variety of names, including also jack-o'-lanterns, friar's lanterns, a hinky punk, and a hobby lantern. And there are many folklore stories about them in English and European folklore also. So essentially what these things were, this is actually methane gas that is released, and then when you put a torch to it, it will light up, it will burn. And it is thought that the moving away of the light as people approach is actually caused by air that is then being disturbed by the approaching person, that would then essentially cause the methane to dissipate, so it'll start to fade away. So the source, there was initially questions about whether the source was bituminous matter, so something like tar or gas. And it was actually George Washington that thought it was a tar-like substance, and it was a Thomas Paine who thought it was a gas. So in this instance, Thomas Paine was actually correct, because it is a gas. There's a nice little video, a link for you to check out about this to give you some more insight. It's like a 4-minute video, but it's pretty interesting. And this is what Thomas Paine said of that event. He essentially said, when the mud at the bottom was disturbed by poles, the air bubbles rose fast, and I saw fire take from General Washington's light. This was demonstrative evidence that setting the river on fire was due to an inflammable air that rose from the mud. And this was methane, actually. Methane gas can cause this. Okay, moving on to the nitrogen cycle. So nitrogen, as we're aware, is required to make proteins and nucleic acids, and N2 actually makes up 80% of the atmosphere. So it must be fixed into organic compounds by microorganisms so that it can be incorporated by animals. So for plants and to assimilate and use nitrogen, it must first be fixed. So that is, it must be taken up and combined with organic compounds. The activities of specific microbes are important for the conversion of nitrogen to usable forms. There are 4 processes that we're going to talk about here. The first would be ammonification, represented by that green over there. So you will start with your decaying organisms. So in ammonification, almost all nitrogen in the soil exists in organic molecules, primarily proteins. When an organism dies, the process of microbial decomposition results in the hydrolytic breakdown of proteins into amino acids. In a process called deamination, the amino groups of amino acids are removed and converted into ammonia, which is N3. This release of ammonia is called ammonification. Next, we'll be looking at nitrification. This is represented by the light green. So when you have your ammonia, what happens now? So the next sequence of the reactions in the nitrogen cycle involves the oxidation of nitrogen in the ammonium ion to produce nitrate, and that process is called nitrification. Now we'll be talking about denitrification. As you can see, that represented by the orange over there. The form of nitrogen resulting from nitrification is fully oxidized and no longer contains any biologically usable energy. However, it can be used as an electron acceptor by microbes metabolizing other organic energy sources in the absence of atmospheric oxygen. This process is called denitrification and can lead to a loss of nitrogen to the atmosphere, especially as nitrogen gas. So again, here you have the release of nitrogen gas, which you can see over there. Now, the final portion, nitrogen fixation. Again, this is binding. You can think of it as being extracted from the air. So essentially, we live at the bottom of an ocean of nitrogen gas. Again, this thing is like, nitrogen gas is like almost 80%, 79% of the air we breathe is nitrogen. And above an acre of soil stands a column of nitrogen weighing about 32,000 tons. But only a few species of bacteria, including cyanobacteria, can use it directly as a nitrogen source. The process by which they convert nitrogen gas into ammonia is called nitrogen fixation. So here you can see the fixation is happening, and then we again end up with ammonia. Okay, so the first thing we're going to be looking at, again here, we're going to, in the same order, ammonification. So this is microbial decomposition that breaks down proteins into amino acids. We have deamination happening. Sorry, I'm going to speed up again a bit, guys. I see I've used quite a bit of time at this one. And the amino groups are removed and converted to converted to ammonia. Ammonification, which is the release of ammonia by fungi and bacteria, and ammonia becomes ammonium ions in water. So here you can see those processes represented in a simple diagram form. Moving on to nitrification, this is the oxidation of ammonia ions to produce nitrate. And this nitrate is then used by plants for protein synthesis. So that's how it gets taken up by your multicellular organisms, your macroscopic animals. And it can also be conducted by autotrophic nitrifying bacteria. And here you see those processes being represented in a diagram form. Then we look at nitrification. This is the oxidation of ammonia ions to produce nitrate. Nitrate is used by plants for specific protein synthesis and conducted by autotrophic nitrifying bacteria. Here you can see those processes from the nitrifying bacteria, but it can also be done by ammonia oxidizing archaea, which would look like this. And then, that will essentially be it for the chemicals and the breakdown we're looking at. So here, what I do want to draw your attention to very, very quickly is looking at the different microhabitats. Here you have the relative abundance of, so we think of them as bacterial species. They aren't really, but just for the sake of argument. So you have your bacterial species being plotted on the y-axis, and you have different habitats, microhabitats being plotted here on the x-axis. So note the community differences stemming from the different habitats. Look how different these columns look from one another. And that gives you some insight into how in which microbes are more successful in which environments. It kind of ties back to what we discussed way at the start of the lecture about how different organisms will outcompete each other and form different niches and be more successful in different habitats. Okay. So then we'll move on to denitrification. Here, nitrate is used as an electron acceptor by microbes in the absence of oxygen. And this produces nitrogen gas, which is lost to the atmosphere. It's typically done by heterotrophic organisms that are facultatively anaerobic. And here you can see a very simple representation of that. Okay. Something that we do need to discuss and that we're very concerned with in terms of the nitrogen cycle as people who are interested in microbiology. Nitrogen fixation is really important because this is a bacterial process that converts nitrogen gas to ammonia. So this takes nitrogen that organisms cannot use, other organisms cannot use, and makes it into a form that other organisms, specifically plants, actually can use. So this is done by the nitrogenase enzyme, and it's carried out by heterotrophic organisms that are usually anaerobic. Then moving on to free-living nitrogen-fixing bacteria. These are typically found in the rhizosphere. So just as a reminder here, the rhizosphere is the region of soil in the vicinity of plant roots in which the chemistry and microbiology is influenced by their growth, respiration, and nutrient exchange. So here are some examples of organisms there, but one that I want to just bring your attention to here is the cyanobacteria that use these heterocysts, which provide anaerobic conditions for fixation. So what is a heterocyst? These are essentially specialized nitrogen-fixing cells that are formed during nitrogen starvation by some filamentous cyanobacteria. So it's pretty interesting. They form these cells when they are deprived of nitrogen, essentially. Okay, and this is just a quick overview of what that cycle looks like without any pretty pictures. So I definitely want you guys to be familiar with all of these processes and familiar with the diagram. The nitrogen cycle is so important. So you should definitely be aware of it. Here you see a split between what happens in the oxygen-rich or oxygen environment, oxic environment, and what happens in the anoxic environment, and where these processes take place and how these molecules change. Okay, now we're moving on to the sulfur cycle. So reduced forms of sulfur, such as H2S, as you can see over here, elemental sulfur, which you can see over there, are energy sources for some microbes under aerobic or anaerobic conditions. Under anaerobic conditions, this sulfur can be used as a substitute for H2O, so water, in photosynthesis by purple and green bacteria to produce elemental sulfur. Something that I do want you guys to be aware here, if you continue on with microbiology, there's a pretty interesting environmental microbiology course that is available at WVU, and there you would do something that's called the Winogradsky column experiment, and there you could actually potentially see the growth of these purple and green bacteria if you're lucky enough, and you will see why they are called green and purple bacteria. Anyway. And then oxidized forms of sulfur, such as sulfates, you can see over there, are used as electron acceptors as a substitute for oxygen under anaerobic conditions by certain bacteria. Many organisms assimilate sulfates to make SH groups of proteins. So important to know. So again, just a bit of a similarity here that I want to point out. The sulfur cycle and the nitrogen cycle actually resemble each other in the sense that they represent numerous oxidation states of these elements. The most reduced forms of sulfur are the sulfides, such as the odorous gas, hydrogen sulfide, the H2S that we were discussing now. Like the ammonium ion of the nitrogen cycle, this is a reduced compound that generally forms under anaerobic conditions. In turn, it represents a source of energy for autotrophic bacteria. These bacteria then convert the reduced sulfur in this hydrogen sulfide into elemental sulfur granules and fully oxidized sulfates. And then just to give you some more background here, that hydrogen sulfide, this H2S, it forms under anaerobic conditions, so typically deeper down in the soil profile, and is a source of energy for autotrophic bacteria. And then it is converted to elemental sulfur granules and sulfates. There are some things that you should be aware of there, that plants then incorporate the sulfates into the amino acids. And a term I want you guys to be aware of here, dissimilation is a protein decomposition that releases hydrogen sulfide into the sulfur cycle. So that's definitely a thing that you should be aware of. Now we'll be discussing the phosphorus cycle. Yeah, so this is another important nutritional element that is part of the biogeochemical cycle. The availability of phosphorus may determine whether plants or other organisms can grow in an area, and there are also problems associated with excess phosphorus. Think about something like eutrophication. We will be discussing some of that later in our remaining 2 lectures, so don't freak out about that too much at the moment. Phosphorus exists primarily as phosphate ions, and undergoes very little changes in its oxidation state. This phosphorus cycle, so there you notice a difference between the phosphorus cycle and the previous 2 cycles we discussed. There's very few changes in its oxidation state in this cycle. The phosphorus cycle instead involves changes from soluble to insoluble forms and from organic to inorganic phosphate. Often this will be related to changes in pH. For example, phosphate in rocks can be solubilized by the acid-producing bacteria such as Theobacillus. Unlike the other cycle, there is no volatile phosphorus-containing product to return phosphorus to the atmosphere in a way that carbon dioxide, nitrogen gas, or sulfide dioxide are returned. Therefore, phosphorus tends to accumulate in the seas. It can be retrieved by mining the above-ground sediments, especially of ancient seas, mostly as deposits of calcium phosphate. Seabirds are actually an example of an organism that mine phosphorus from the sea by eating phosphorus-containing fish and depositing it as guano or bird droppings. Just a brief summary of this phosphorus cycle again. Phosphorus, we don't have those many reduced or oxidized forms. You have changes from soluble to insoluble forms and from organic to inorganic forms. There are no volatile forms where this phosphorus is returned to the atmosphere. Thus, phosphorus tends to accumulate in seas. It exists primarily as phosphate ions and is related to pH. Here you can see that Acidotheobacillus that produces acid that solubilizes phosphate in rocks. Okay, moving on. For our last couple of slides, we're going to be at least now we're moving on to microbial bioremediation. So what is bioremediation? So that is, in this case, microbial bioremediation refers to the use of microbes to detoxify or degrade pollutants. So examples here, you can look at something like oil spills from wrecked tankers and drilling accidents, where microbes can be used to help clean up those forms of chemical pollution. This is actually a picture taken from, I think this was the Deepwater Horizon spill. I think so. So something that is really interesting though is that bioremediation is actually, it occurs naturally as microbes attack the petroleum if the conditions are aerobic. However, microbes usually obtain their nutrients in aqueous solution, and oil-based products are relatively non-soluble, so that creates a problem. Additionally, petroleum hydrocarbons are also deficient of essential elements such as nitrogen and phosphorus, so they lack nutrients. Therefore, bioremediation of oil spills is greatly enhanced if the resident bacteria are provided with essentially a fertilizer. You can think of it as a fertilizer containing nitrogen and phosphorus. So something that I definitely want to point out, if it wasn't clear at this point, the one thing that is really helpful here, or that we're exploiting here, is the fact that you can find almost any microbe anywhere. Microbes, there was a famous quote from a microbiologist who used to say, everything is everywhere, then the environment simply selects. We know now that is not 100% factual. A more corrected statement of that would be that some things are most places and the environment selects. But be that as it may, a nice advantage of using microbes in these circumstances is that they are typically already on site. So it's not that you have to take in tons of microbes. You don't have to grow them in a lab somewhere and bring them in. And then you typically, if you produce the environments for these microbes to thrive in, they will thrive. So they will clean up the mess, and they're already present on site. So that's big benefits, something that is really beneficial for bioremediation. Anyway, moving on to maybe a problem that you guys will be more aware of because you live in, or at least you study now in WVU. So acid mine drainage is a big problem in this state. So over 2,000 miles in West Virginia are contaminated by acid mine drainage, resulting in acidic water and high concentrations of metals that prevent aquatic organisms from inhabiting these ecosystems. Typically, the prevention happens because this decreases the pH, and most organisms just simply cannot live in this environment. And there are just high concentrations of metals and sulfates that are also can also be harmful. And so microbes can be used or there have been proposals at least, and some of the microbes can be used to help with this problem of acid mine drainage. So looking at that, if you look at wetlands, for example, you can the anaerobic microorganisms in wetlands can be used to help remove sulfates, which will lower the pH and iron from the contaminated waters. Processing of these compounds relies on a variety of microbial processes, so it's pretty complex. So first thing I want to ask you guys, how do wetland construction techniques affect these processes? That's something that you have to take into consideration. How you build or how you manage your wetland will affect its efficacy to a large degree. And then on the same vein, you have to think to yourself, what can we do to help improve these populations, or help grow these populations that will help reduce the acid mine drainage and to effectively remove these contaminants? You kind of have to ask yourself, how can you manipulate the system or manipulate the ecology to our advantage without causing catastrophic harm? Okay, so first, again, we will start with your complex organic matter. These wetlands are typically rich in complex organic matter, and this will result in, firstly, you will have your monomers and oligomers here, and they are released when the complex soil organic matter are utilized by fermenting organisms, and this will release simple organic compounds such as waste. So here you have simple organic compounds that are expelled as waste from fermenters and from the acetogens. Okay, so starting at that point, now you will have these compounds are then consumed by bacteria that use iron and sulfate as terminal electron acceptors, thereby reducing them. So here you have iron reducers, here you have your SO4 reducers that will take in the compounds like this is our water and this is in the water because of the acid mine drainage, and we don't want them there. So we have to get to a point where we can produce or can think of almost upregulates the wrong word. You can create conditions favorable to these iron reducers and the sulfate reducers. And then, so once that happens, once the reduced sulfate can abiotically join with the ferrous iron, so over that, and precipitate in the soil, for instance, through the formation of pyrite. The reduced sulfur can then also leave the system gaseously via the formation of hydrogen sulfide gas or dimethyl sulfide. You can see that, the hydrogen sulfide and the dimethyl sulfide. Due to these processes, wetlands are often constructed for the treatment of contaminated waters. But as you can see, this is a complex system, and many questions remain with regard to how different wetland construction approaches affect the development and activity of these microbial populations. So again, this entire process hinges upon these iron reducers and these sulfate reducers. So you have to create an environment where they are as effective as possible and are given the opportunity to remove the contaminants from the water. So I mean, you can construct your wetlands in different ways to try and maximize or optimize this process, increasing its efficacy, obviously. Okay, so that will be it for the lecture. And for today's review question, I decided let's use an example of a longer question. This is something that might be on the final exam. So here my question I'm going to ask you, why is the diversity and population numbers of microbes per unit volume of soil typically greater than in a similar volume of water? I've already used a lot of time here, so I'm actually going to, if you want to think about this answer yourself, or you want to write up an answer, this can count anything between 5 to 10 marks, depending on how much detail I want. But pause here because I'm going to go on to the answer immediately, or the discussion of the answer immediately. So I just put down a few bullet points here that would pertain to the to the answer. So firstly, I would definitely want you to mention microhabitats and the different niches that these organisms can inhabit, and how there would be more more of certain organisms in certain portions of the soil than in others. And this is because the different organisms can compete most successfully in different areas of the soil. And the soil, the fact that soil can create these microhabitats, or these niches that need to be filled by the microbe, is exactly why, or is one of the reasons why you have greater diversity, because water is far more homogeneous. If you look at a water sample, with diffusion happening, they're like, especially if you look at just because I said per unit volume, so any volume of similar water would be far more homogeneous in terms of nutrients, temperature, moisture content, obviously. Typically would be far more homogeneous, and that the homogeneity will lead to a lack of microhabitats, a lack of different microhabitats. So if you have a certain volume of water, you will have microbes that essentially will outcompete the others, and those will be the ones, and they could outcompete those other microbes completely into oblivion, into extinction, so that they are the only ones that occur, or at least the far dominant ones that occur. Microbes can also obviously move around in water way more easily than they can on, in a soil clump. So, if there is a competition for resources, the ones that are outcompeting the others would be, that effect would just be multiplied. Then I also want you to know that typically there's a lack of nutrients in environmental water sources, especially open water, where compared to soils. Soils, I mean, there's how much organic matter can you find in soil, in topsoil? If you look at something like a forested soil, yes, there are examples of soils that are not very rich in organic matter, but more often than not, typically at least, you will find that water sources will lack nutrients when compared to soils, because soils retain nutrients a lot better. So this will provide the microbes with more food sources, which will also lead to increased numbers. And then obviously the lack of nutrients will also lead to less opportunity for rapid growth. So even if you were to newly inoculate water samples and soil samples, the nutrient differences might also lead to a problem. So these are the type of things I want you to think along with. When you find a question like this, you have to think about the media that you're working with, how microbes will behave in that media, how they will be affected by the media, and the constraints of the media, and why would that lead to greater diversity and population numbers in the soil than compared to the water. Okay, guys, I hope that helps you for the exam, and I will then see you in the next lecture. Okay, guys, cheers.